Hey everyone, welcome to this episode of Genomify. This will be the first video in the Cut and Run data analysis series, and today we will be covering the alignment and normalization factor calculation for raw cut and run sequencing data. And yeah, again, like the RNA sequencing quantification video, we'll be using DRAGEN. And so just kind of walking through what we'll actually be covering in this video. Um, First, like the RNA quantification video I previously posted, we're going to be walking through how to launch a DRAGEN instance on AWS. Second, I'm going to show you how to create a hash table, in this case for the E. coli genome, but please reference the RNA sequence alignment and quantification video for the human hash table creation. Second, we'll be showing how to actually perform this mapping and aligning of this raw cut and run sequencing DNA data. And within this, something that's maybe a little unique to this video will be that we are both removing duplicates and trimming adapters. And for those of you who aren't familiar with what that exactly means, so I guess I'll start with adapter trimming first, actually. So in a sequencing library, you kind of have these adapters that are the same throughout the library that are attached on either end of your DNA inserts. And this is just used to attach indices, and this can separate samples. So when you load a ton of samples on a flow cell, you can actually disaggregate them once the data is read out. So we have to cut those off though. And DRAGEN's really good at doing that. And secondly, we'll be doing duplicate removal. And for those of you who are not familiar with this, during library preparation, you amplify up your initial library, and this causes certain fragments of DNA to be essentially duplicates of each other. And so you're taking a very, very tiny portion of this PCR amplified library and sequencing it, but some of the fragments within that sequenced batch end up being duplicates. So you have to filter those out. And those are two things that'll be different in this video as compared to the RNA-seq alignment and quantification video. Um, Again, just to highlight, we'll be using Illumina DRAGEN. And lastly, probably one of the more confusing aspects of cut and run that's pretty unique to it is we'll be doing this E. coli normalization factor calculation, and I'll walk through both mathematically how I do it and use a cool little metaphor to help you understand why we're doing it and what it actually means. So just some requirements slash what kits I use to do this. We'll be needing access to AWS and in particular the DRAGEN software. And if you have prepared your own samples, what I did, and I know it'll work for this particular analysis, is I used the EpiCypher Cut and Run Katana Kit version 5 and the EpiCypher Library Preparation Kit version 1.5. The library prep kit version, I just know that the adapter sequences are on the DRAGEN instance. I don't know about other kits. I would imagine they probably are, but just a warning. And so just to kind of walk you through before I jump into the data analysis of what I'm actually looking at. So in this exact experiment, I had sequenced, I believe, 32 different samples. It could have been more, but it was a lot of different samples. And we worked with Novogene and we bought a full lane on a sequencing chip. And what we did is we asked for around 5 gigabases or 5 billion nucleotide bases per sample. And we found that this was sufficient to detect realistic binding profiles and actually perform differential chromatin binding assays. So just to kind of give some recommendations for what may be useful in your experiments. This ended up being around 15 to 25 paired end million reads, 15 to 25 million paired end reads per sample, or around 30 to 50 million reads overall. And that'll kind of be able to be disaggregated once you perform alignment. We ran these in biological duplicates, and all the samples were run in one sequencing batch, except there was one that we had to run again and had to do some downsampling. And I might make a video later just showing you how you can perform that downsampling if something similar happens to you. Okay, so you just did your cut and run experiment and you have your raw sequencing data back. The first step we're going to need to do is align that DNA to two different reference genomes, both the human and the E. coli reference genome, because we use that E. coli spike in DNA to perform that normalization. And so the first thing we're going to need to do is align this DNA using a software called DRAGEN. There are other ways to do alignment. However, this is what my lab uses. It's super quick and we like using it. So I'm going to show you how I launched this um, kind of software and how we perform the alignments. So we'll just type in DRAGEN to the search bar, click View Purchase Options. We'll come here, click Launch Software. And then we'll do this Amazon EC2 and launch from EC2 console. That gives us a little bit more customizable options here. And so we're just going to use this most recent version. Um, an annoying part about DRAGEN is the versionings get deprecated, so you may need to update this analysis if you're watching this way in the future, but nonetheless, that's the version we're going to be using today. And so we'll click Launch from EC2. And so I'm going to be walking through within like the Emory system, and so this will be particularly applicable to Hung Lab members, but I'm assuming you have some familiarity with how you launch AWS instances if you're watching this from outside of Emory. So it's going to pick a recommended compute size, in this case the F2 6x large. I will pick my key pair that I'll use to connect. This is specific to Emory and the Hong Lab, but you're going to want to select Emory IP. I like to bump up the storage to around 200 gigabytes. I'm only going to be analyzing one sample here, but if you're doing numerous samples, for instance, this analysis actually did like 32 samples at once, you may want to bump that up to around a terabyte. And then again, for the Hong Lab members, you're going to want to change this IAM instance profile to EC2 to S3. This will just let you move files between the S3 and your DRAGEN instance. And so other than that, everything stays the same. And we'll click Launch. And so we'll click that, and that'll take us to our instances. One note here, if you're logging in on Emory, you're going to have to be connected to this big IP VPN, or it will not let you connect. However, so it seems to be launching already. And so what we'll do is we'll copy this SSH command and let me log in here. So you're going to need to navigate to the folder that has your PEM key. So in this case, my G Cooper Hung Lab PEM key is right here. I'll copy that SSH command into the command line. We actually delete the recommended IP address here. At least for Emory, you have to copy this private IP4 address. I don't know why, but that's the way it is. And we also are going to have to change this to EC2 user. Let's see, why are we getting there? Sometimes it takes a second to actually launch. There we go. So click yes, and that'll just take us into the ephemeral instance. And once you see this EC2 user pop up, that means you're actually in this remote machine now. So we're going to have to navigate up to this ephemeral directory. That's where all the actual compute and storage is held in. Okay, so we launched our instance. Now we need to download our reference genomes and create hash tables. And so I'm going to show you how to do this for the E. coli genome first, but I'm assuming you can follow along to my previous video looking at RNA-seq alignment, and you can do the same for the human genome. So I'll show you how to download this E. coli reference genome and how we create the hash table for it. So to download the E. coli hash or E. coli reference genome, you need to navigate to this iGenomes. Illumina has a lot of reference genomes already kind of pre-deposited into this database. And at least if you're using EpiCypher version 5 kit, this is the spike in E. coli DNA you'll be using. So if you just click this link, it will download a folder with multiple files in it, but that will contain the FASTA file. I already actually uploaded this to my S3 instance. And so I'm just going to move along as if you've already done that. So what I'll do is navigate to my S3. We'll go to where we have that particular. So this is what we'll download when you click that link. There's going to be a couple nested folders you have to navigate through. Click NCBI, click this date, click sequence, click whole genome FASTA, and then click this E. coli genome FA. Copy the S3 URI. Then you're going to want to copy that FASTA file to your DRAGEN instance. Let that cook for a second. Okay. And so then what we're going to need to do is create the hash reference, which is a file format that DRAGEN uses to perform alignment very quickly. And so I'm just going to make a directory called mg1655, and this will be the hash version 11. This is kind of just terminology within DRAGEN. But ultimately, you can save this for the future. If you just want to pull this hash graph directly onto your DRAGEN instance, you won't have to redo all this whole process. So I'll make that directory. I'll move the reference FASTA into that directory. Okay, and so now we have that in there. And so now we're just going to tell DRAGEN to make this hash ref folder system. So we'll just call it DRAGEN. Say we want to build a hash ref or hash table, and we'll just put true. And then we'll just tell it where the reference is. And so in this case, it'll be, and then E. coli genome.fa. And then we'll tell it where to save everything. And in this case, we're just going to write everything into that same folder we just made. All right, so this should take no more than 10 seconds or so. I will note, if you're doing this with the human genome, it actually will take quite a bit of time because it's a lot bigger. But luckily for us, E. coli is pretty tiny in terms of genome size, so we'll let this do its thing. And so while we're waiting, though, I will say, like I said, you should move this onto your S3 to save as kind of your backup reference. It's annoying to have to do this every single time, so just save it on your S3. I've already done this for the human, and every time we do these alignments, we just move that right back onto our DRAGEN instance, so we can just move right into the alignment process. So yeah, it completed, and so it has a lot of different files. This all just helps DRAGEN align things more quickly, and so the same idea will be applied to the human reference. And like I said, if you go reference my RNA-seq DRAGEN alignment video, that will show you how to do this for the human genome. But I'm going to move everything onto this instance, including the raw sequencing data as well as the code, and I'll kind of walk through it in a bit. But I don't want you to just watch me move files around. So I will see you in a bit after I finish doing that. Okay, so I just moved everything that is needed for this analysis onto the same DRAGEN instance. And just kind of walk you through what I actually did. I made these two empty output folders called E. coli alignments. Oh, I need to make one more called human alignments. And also, I pulled in this human genome hash table. And I also brought in our raw sequencing data for only one sample here, but this analysis pipeline can be applied to multiple samples at once if you want. And so here I'll show you the code that we'll actually be using to run this alignment with DRAGEN, and I'll walk you through each one. And you can find this both in the video description as well as my GitHub I'll provide as a resource. And so just kind of walking you through here what we're going to do. So like I said earlier, we're going to align our sequencing data to two different reference genomes, the human and the E. coli. And so each of these text blocks here is telling DRAGEN to align it to each different reference genome. And so for the top one, we're telling it to align it to the human genome here, and we're telling it to align it to the E. coli genome here. Then what we're doing is just providing the raw sequencing data files. And then another thing that is pretty unique, and I did not include in the RNA-seq alignment video, is that we need to trim the adapters that are used to attach indices onto each of these DNA library inserts. And so luckily DRAGEN has these pre-made adapter sequence.fasta files, and it just pulls that into the command and trims those off of all your reads. Again, also something different here is that we're actually going to be removing the duplicates that arise during PCR um, cycling of library preparation. And then we'll just tell it where we want to save all the outputs and all these prefix things that are weirdly specific to DRAGEN. And this is also something you will need to include if you're using this particular version of DRAGEN 4.4.4. And so yeah, we'll basically just run this command. And just a quick note, I had to make this. This is just a text file I made with the vi. So I just did like vi test or whatever. And so you can just copy paste whatever you want in there. And then I made it accessible with this trim_odd plus X, and then I did code.sh. And so that just makes it so you can execute that command. And so then that'll run and it'll output all of our outputs into the E. coli alignments and human alignments folders. So the way that I run this, particularly important if you're doing a lot of samples at once, is this no-hut command, /thing. It's in this folder, and we're going to run this command here. And then also use ampersand just so I can assess the output while this is all running. And so this will probably take around maybe 3 to 4 minutes to run. So I'm going to let it do its thing, and I will be back when it is finished. Okay, so the alignment's finished as I thought. It took about 4-ish minutes. And so if you navigate into the actual output folders, you can see that you get your BAM file or your aligned fragment file, as well as the necessary indices. And you also get a lot of these kind of summary metrics files. And so while I typically don't dig too deeply into these, except just to make sure I'm not getting a lot of a high unalignment rate, for cut and run, you actually do need to use these quite in depth. So we're going to use the mapping metrics for both the human alignment as well as the E. coli alignment to perform sample normalization. And so I'm going to walk you through here how we're going to use these mapping metrics to perform sample normalization. So what you're going to do is you're going to look into this mapping metrics file. We're going to pull out some key metrics that are going to be useful for calculating that normalization factor. And so let me get rid of that. Get rid of that. So I will pull up this. So this is kind of my full sequencing metrics file. This is what I had done for my manuscript for SMART-One cut and run assays. And here I'm just showing you the analysis for this one sample, but as you can see, I had a lot of samples for this project. So the important ones that we're going to need to pull out from this mapping metrics are going to be the unique reads. And this one I kind of pull out more just for intellectual curiosity. We don't actually need it for the normalization factor, but also this, we will need this unique human aligned metric. And so that one's actually not going to be in this output because this newer version of DRAGEN just doesn't do it. So we're going to have to do some kind of back calculations to get what a previously older version of DRAGEN outputted. So I'll show you how to do that. So what we'll do is we'll come back here, and I copied over the total reads just to kind of have as a background metric and report to the lab on how the study went. The other thing you'll need is the number of unique reads. And so that one I just copied right over here. And then here's the number of mapped reads. And this is the kind of tricky calculation you're going to need to do. So we need the number of unique mapped reads to perform the normalization factor calculation. And so the way that we're going to get this unique mapped reads is we're going to subtract from the unique number of reads the number of unmapped reads. So I'll copy that over here. And so in this case, we're just doing unique reads minus the number of unmapped reads. And so the reason we're doing this is because the unique reads include everything, including both reads that mapped and those that didn't map. And so when we subtract the unmapped reads unmapped reads, we're left with only the unique reads that were successfully aligned to our reference genome. So it's kind of annoying. Previous versions of DRAGEN just gave it to you as a outputted metric, but we have to do this weird math to calculate it this time in the newer version. So you'll encounter this when you use DRAGEN. But nonetheless, we're really only going to pull two values out from this file, and yeah, that'll give you basically the number of unique mapped reads. And so we'll come back here and jump into the E. coli output. And as you can see, we have a very similar set of outputs here, including the alignment file as well as the index. But really for the E. coli alignment, we're only going to be looking at this mapping metrics. And again, so we'll just pull out number of unique reads and then subtract out the number of unmapped reads, and that will ultimately give us the number of unique mapped E. coli reads. Okay, and so once you've pulled out all these mapping metrics for all your samples, and it's, as you can see, this actually took me quite a long time. A lot of things to be copying and pasting. You got to be very meticulous, and when you do this, make sure you don't make any mistakes. And so I'm sure there are better ways to do this, but this is what I did. So once you have all this, though, you will then calculate this E. coli bandwidth, which is essentially a ratio of the E. coli compared to the unique human aligned DNA. From this bandwidth factor, you will then do 1 over the E. coli bandwidth, and that'll kind of give you the scaling factor that you will then multiply both your bed graph files as well as your bigwig files. So at least within CUT&RUN, what I did is I used the scaling factor. Oh, let me get rid of that. What I did is I multiplied my bed graph files prior to peak calling just to minimize IgG signal, because as you can see, there's a lot more E. coli DNA proportionally to the actual antibody target DNA samples. And so you're basically kind of like shrinking the signal of that IgG so that it is a background and it's not kind of overwhelming the samples that were actually eluting DNA with antibody binding and cleavage. And so, yeah, I'm going to explain this with an analogy just a sec. I kind of need to think about it a bit. So give me 1 second. Okay, so I'm going to try to explain this coli normalization with a metaphor to hopefully make it a little bit easier to understand. So the issue with CUT&RUN is that you're not actually measuring biological differences across different samples based on just raw read counts. You're also taking into account technical differences between each sample, particularly the amount of starting input DNA. So for instance, the IgG control has not that much starting input DNA into library prep. You typically get under a nanogram or less. For a mark like H3K27 trimethylation, you get a ton of starting material, up to like 30 nanograms. And you ultimately load 5 nanograms into the library prep, but this is just a very drastic difference of amount of diluted DNA. And so imagine you're a baker and you're trying to compare how many chocolate chips are in different batches of cookies. However, each batch has a different amount of starting material or dough. And so the way that a baker can then try to assess different numbers of chocolate chips in a baking exercise would be to add the same amount of raisins to each batch of cookie dough. And so the idea is if you're starting with a ton of cookie dough and you add like 10 raisins, at the end, if you're sampling 5 cookies, you're not going to get that many raisins because you had a ton of dough to start with. However, if you add the same amount of raisins and sample to a smaller batch and then sample those another 5 cookies from the smaller batch, you're going to get more raisins per cookie. And so this is essentially the same thing we're doing with CUT&RUN. Instead of raisins in this case, we're going to be using something like E. coli DNA. And so then each sample, irregardless of how much starting DNA material is being put into library prep, they'll all be compared relative to the amount of raisins or amount of E. coli DNA that is included in each sample. And so, based on this kind of raisin metaphor, that'll give you this E. coli bandwidth. And so, assuming you have a smaller batch, you're going to get a higher proportion of cookies with raisin than you would with a bigger batch. And so that's kind of why we're getting this high E. coli bandwidth for our IgG samples, because you're not starting out with that much starting material. You're adding the same amount of E. coli DNA, so you're going to get a higher ratio of the unique E. coli aligned compared to the unique human aligned. So the thing is, though, when you just sequence these samples, they're all going to look at least like kind of similar. The IgG will be lower. But the thing is, you need to shrink it even further because realistically, you're getting the same kind of number of reads from each sample, but because IgG had so much E. coli present, you know that it had a very, very tiny starting amount. And so then the way that you calculate the scaling factor is this 1 over the bandwidth. And so essentially, samples that had a high E. coli bandwidth are going to get shrunk more, meaning that the each value from the bigwigs or the bed graphs, and we'll come to this later, is going to get shrunk. However, samples that had a ton of starting material are going to be kind of artificially inflated because the E. coli proportion is telling us that there was a ton of starting material. So we kind of have to inflate that up, and that will kind of give us a more realistic biological representation of how much DNA was actually being eluted during the cut and run experiment. So essentially, you could think of it also kind of like speakers. So imagine each of these samples is like a speaker. The IgG, it's being really artificially loud. In comparison, this H3K or 27 trimethyl is being really quiet. But to really normalize them, we need to turn up the H3K27 to actually represent what's going on here. And for the IgG, it's artificially loud. So we have to kind of turn that one down. And so that's kind of what we're doing here. And ultimately, we'll use these scaling factors to multiply both the bigwigs for visualization purposes as well as the bed graph files for peak calling. And so we'll kind of get to that in a later video, but I just wanted to kind of explain why we're doing this. I know when I was first starting out, it was really hard for me to understand why you even wanted to perform normalization on a cut and run experiment like this. So yeah, with that, that is pretty much the end of this part of the video. What I would recommend, oh, it logged me out of here, but what I would recommend is saving the BAM files only for the human alignments here, as well as the indexed BAM file as well. These will be important for downstream processes. And I would not recommend keeping the BAM file for the E. coli. It's actually not that important. You really only need to get these mapping metrics from it. But I would save these mapping metrics as well as trimmer metrics. And I think those are the only two that are going to be important. Maybe this fragment length distribution just to get an idea of how your library was for different QC metrics. But oh yeah, I think it would also be a good idea to save this fast QC metric so you don't have to rerun all this analysis there later down the line. So yeah, you can just move these files onto your S3, keep them for future analyses. And if you ever need to redo this sequencing metric thing, if you think you made a mistake, you can always come back to this mapping metrics file and kind of double check yourself. And so yeah, that's pretty much the end of the alignment video. And in future videos, I'll show you how you actually use this scaling factor to multiply different files and ultimately perform peak calling and graphical visualization. So that's the end of this episode of Cut and Run data analysis. Stay tuned for more. I hope you learned. Please leave comments, questions if you are not understanding anything that I showed you. And as always, please like and subscribe, and I will see you next time.